Good morning, good afternoon, good evening, depending on wherever you are. Welcome to session number 18 of our ASLM special sessions on COVID-19, a series that we have dedicated to diagnostics in the COVID-19 pandemic. Today, we are hosting an exciting topic. We're going to be discussing a topic around serology. And as a second time, I think in the history of our series, I think it's also going to be hosted by our colleagues from the private sector. Today we have Roche International Limited, who are going to talk about what they are offering in terms of serology coming through. We all acknowledge that there is still quite a lot to learn from serology as we move on with the pandemic. My name is Anafi Mataka from ASLM, and I'll be your host today. I also want to give you a few logistics notes as we move on. As usual, we will start with a presentation for about 20 to 25 minutes, followed by a question and answer segment. Please enter your questions or comments into the chat box, and we shall address them one by one after we finish our presentation. I also want to inform you that we now have language interpretation from English to French, so feel free to tune in to the language of your preference, either English or French. To take us through our presentations today, two distinguished gentlemen from Roche, in the name of Dr. Wim van der Helm and Dr. Matthias Strobbe. Dr. Wim is head of healthcare development for Roche Diagnostic region for Europe, Middle East, Africa, and Latin America, which they usually call it LATAM. This role includes affiliate support in the implementation of Roche scientific and medical tactics, the Medical Speaker Bureau, and the partnership with key external stakeholders. He holds an MD from the University of Berlin and an MBA from two academic institutions in Germany and France. Before he joined Roche in 2004, Wim had held several positions as a practicing physician in the Medical Relief Organization, MSF, Médecins Sans Frontières. He is a member of the Diagnostic Medical Leadership Team. And Dr. Strobbe is a senior clinical science leader for infectious diseases for Roche Diagnostics. He trained as a microbiologist before turning his attention to virology during his PhD thesis. After leaving the academia for industry, he undertook several clinical trials in the infectious disease field, evaluating drugs against invasive mycosis, HIV, and HCV. Dr. Strobbe joined Roche Diagnostics in the medical and scientific affairs team, where his main responsibilities are implementation of clinical strategy for infectious diseases as part of their infectious disease team. And he also provides medical and scientific expertise with a particular focus on studies and providing input for product development, such as the ECLIS anti-SARS COVID virus 2 that we're going to be looking at today. Ladies and gentlemen, let me hand over to Dr. Wim and Dr. Matthias to take us through. Over to you, gentlemen.
Okay, thank you very much, Anafi. Can you hear me okay and see the slides?
Yes.
Perfect. Okay. All right. Welcome, everybody. Good afternoon. Thank you very much for allowing us the opportunity to share some of the data and considerations from Roche Diagnostics with you. I would like to get started with honoring a colleague of ours. And it's not working at the moment. 1 second. Here we go. And that is Dr. Li Wenliang, who was one of the first healthcare professionals that actually recognized a new virus in his patients. Within weeks of the first cases, the first NUT tests, specifically real-time PCR tests, were developed by laboratories worldwide. And this, of course, was possible because of the dramatic achievement of quickly sequencing of the genetic material of SARS-CoV-2, which was then a very new virus to humans. Now, what we do today, all statements that we make in this presentation are based on the current state of scientific literature. As we all know, the field is evolving quite fast, and we see almost weekly new evidence coming up. However, what we share with you today is what we know today, and there is definitely more evidence to come. And I encourage you all, together with us, to help develop this evidence. Diagnostic tests are an essential service in the delivery of healthcare, and in vitro diagnostic testing has become an indispensable tool in clinical practice. It can provide critical information at every step of the patient pathway. So results in vitro testing influence as many as 70% of clinical decisions. Never has this been more true as for the COVID-19 pandemic, since it has opened a new window into the world of diagnostic testing. COVID-19 has disrupted lives of billions of people, nearly overwhelming healthcare systems and pushing the global economy to the brink of collapse. So the quest for effective treatments and preventive vaccines starts with and depends on the broad availability of fast, accurate, and reliable testing. IVD testing plays a vital role in testing who may have contracted COVID-19, ensuring that healthcare professionals can work safely and that quality care and treatment is available to all those who need it. And helping to flatten the curve, lowering the rate of spread of infection, providing governments and healthcare institutions with actionable test results. So high quality diagnostics aid more accurate disease identification, is leading to more appropriate patient care. The two primary important questions when using IVD testing for COVID-19 are: the first one, is the patient shedding virus and potentially infect other people? And the second, has the patient recovered from a previous infection and developed antibody response to the virus? It should be noted that currently there is no evidence that people who have recovered from COVID-19 and have antibodies are protected from a second infection. However, to contextualize this, the reports that we see from patients that supposedly have had a second infection is a handful of patients, and it's quite questionable whether these patients actually have been reinfected or they have been maintained infectious. So it's not really robust evidence that is suggesting you can get reinfected with COVID-19. Coronaviruses are zoonotic, which means they are transmitted between animals and humans. And the animal reservoir is ecologically diverse with the widest variety seen in bats, which are the reservoir for many of the other viruses. Mammals may serve as intermediate hosts, facilitate recombination and mutation events with expansion of the genetic diversity. There are currently 7 known human coronaviruses with varying harm factors. We know that there are 4 endemic common cold viruses, which are responsible for 5 to 10% of all mild upper and lower respiratory tract infections, and 3 severe respiratory syndromes with high fatality rate. And those are SARS-CoV, SARS-CoV-2, and MERS-CoV-2. We've seen 2 previous outbreaks in China and in Saudi Arabia. Now, what are the signs and symptoms for COVID infection? Transmission, as we have seen in other presentations as well, is person to person via respiratory secretion. Incubation time is ranging between 2 and 14 days. The main symptoms are fever, respiratory syndromes, and actually the most common is anosmia. The clinical presentation is ranging from asymptomatic infection to mild illness, pneumonia, as well as fatal disease. And when there is clinical progression, that can cause the severe respiratory disease, especially in patients aged 65 and older. The case fatality rate is quite varying, and at the moment we have data that it's ranging between 0.8 and 15%. Very quickly, risk factors for severe disease and death. As we said already, older age, male sex, comorbidities, specifically hypertension, diabetes, cardiovascular diseases, obesity is a risk factor, and then tobacco consumption. So what are coronaviruses? These are relatively large viruses compared to other viruses we know. They have a diameter of up to 120 nanometers. They are enveloped RNA viruses with a single-stranded RNA genome, and the RNA is largely covered with a protein called the nucleocapsid protein. We will hear more about this particular protein when we introduce our Alexis assay. The nucleocapsid protein is one of the 4 structural proteins of coronavirus. The others are the membrane protein, the envelope protein, and the spike protein. Infection path, the virus enters the patient via airborne droplets. The incubation phase between 2 and 14 days. However, typical is 5 days, and the infected patient can then transmit the infection either by respiratory secretion or by contaminated surfaces. Once the virus enters the body of the patient, viral replication begins, which in turn presents in a wide range of symptoms in the patients. It is important to note here that clinical presentation can vary from patient to patient, with some patients or many patients actually being asymptomatic. As with clinical presentation, clinical progression of the infection is varied but tends to be more severe in older and compromised patients. After the virus enters the body and reproduces, the immune response is triggered and antibodies are formed. During this period, the patient is deemed infectious and can pass on the infection. Once the virus is eliminated and neutralized by either neutralizing antibodies or cellular immune response, the patient recovers. And again here, it is very important to note that even if small amounts of virus are present along with antibody response, the patient may still be infectious. With each phase of the infection, typical clinical symptoms appear. These clinical symptoms can be evaluated using a range of diagnostic throughout each phase as the infection progresses. And if we look at the disease progression, there are 2 main phases. The first one is called the viral response phase. In this phase, the virus itself causes the disease symptoms by infecting cells and lying those cells during proliferation. This then may move over into host inflammatory response phase, which is characterized by the body responding to the infection. In roughly 5% of the cases, it can lead to hyperinflammation, which is in the end what causes the severe symptoms and can eventually lead to death. Now, a number of health protection agencies have issued testing recommendations for governments, healthcare systems, and IVD suppliers as guidance. And we at Roche take these guidance into consideration during test development so that we launch solutions that meet actually these requirements. Now, let's look into the types of markers and their development throughout the course of the infection. In ongoing infection stages, viral RNA and antigen appear usually quite quickly and have a steady decline to convalescence. So measured viral RNA and antigen are indicative of infection and infectivity. Once the immune response begins and antibodies to the virus start to develop, the levels of RNA and antigen drop significantly. The first antibodies to appear are IgM, closely followed by IgG, and seroconversion takes place around the medium of day 12. The infection is then said to be resolved and potential immunity is gained. However, more studies are required to understand the longer-term antibody profile and immunity levels of the patients. Specifically in SARS-CoV-2, the antibody actually patterns quite antibody patterns differ quite from textbook patterns. So we will also see later in this presentation the dynamics of IgM and IgG, where IgM and IgG almost simultaneously raise. Now let's look into the types of markers and their development throughout the course of the infection. That's the response to infection. When we look at the molecular course of serology biomarkers in relation to what we are trying to find out in diagnostics, we provide two types of IVD tests. The first one is to directly detect the virus currently circulating in the body. Therefore, high quality PCR testing for SARS-CoV-2 is used. And the second group of tests, which we will discuss in more detail later on in this presentation, is analysis of immune response and resolved infection. So therefore, immunoassays can detect immune response to SARS-CoV-2 antibodies. Now, detecting the response to virus is essential and has many implications for patients, healthcare services, and governments. Once immune response begins, antibodies to the virus start to develop, and the measurement of these antibodies provide insight into true exposure in a patient or a patient population. The development of antibodies to the infection usually is seen in both immunocompromised as well as immunocompetent individuals. However, immunocompromised individuals show a stronger response to antibodies. In the case of COVID-19, the detection window for antibodies is longer than the detection window for direct virus detection. Now, what is of critical importance is also what features an assay must have and what a false assay result would mean for the patient, which is finally the medical risk for the patients. Antigen assays and PCR need a high sensitivity. This means they must be very reliable in detecting the presence of the virus, since false negative and missed cases can lead to a high risk for the infected patients and for further spread the virus to others. That's a bit different for antibody assays. Antibody assays need to have a high specificity, which means they must be very precise in telling that a negative test is truly negative, since false positive results lead to the erroneous assumption of convalescence and putative immunity and putting the person at risk of infection by otherwise preventable exposure. The Elecsys anti-SARS-CoV-2 assay, which we will hear a bit more in detail at the next talk, is intended as an immunoassay for the in vitro qualitative detection of antibodies to severe acute respiratory syndrome, coronavirus 2, in human serum and plasma. And the test is intended as an aid in the determination of immune reaction to SARS-CoV-2. I would also like to share with you links to educational and technical material. We have two dedicated websites in which you can retrieve information about the individual PCR test as well as the antibody test. I have provided the links on the bottom in this slide deck, and I will share later on also with Anafi, who can forward them to you guys. With this, I close my talk and would like to lead over to my colleague Matthias Strobel, who will go into more of the technical performance details.
Thank you, Wim. And also thank you, Anafi, for this kind of introduction. I'm just trying to give you access to my screen. Here we go. Yes. Can you all see my screen?
Yes, it's clear. Thanks. Go ahead.
Thank you. So it's a great pleasure for me to introduce you to the Elecsys anti-SARS-CoV-2 assay. that we have brought to the market sometime in early May. This was a record time for us. And I also would like to give you some background why we designed the assay as it is now, and as it is now also being used in labs all over the world in the meantime. So first of all, let me start with providing you with a rationale for the selection of the test format, and also have a look at the antibody response first. In the introduction, Wim already mentioned the viral structure with the different viral proteins. So the question for us was, what type of antibodies are being generated that we could or should target? Therefore, there are clearly antibodies developed against the spike protein, what we know, which are called the anti-S antibodies, and then also antibodies against the nucleocapsid, which in turn are called the anti-S antibodies, what you can see on that slide here. It's in the structure in the RNA, binding to the RNA, that's the red structure in the middle. And as Wim already mentioned, in contrast to what we know from other pathogens, the serological profile here shows a great variability. This probably also comes from the endemic coronavirus that are already circulating and cause a common cold. And this may be one of the reasons it is being speculated that we don't see a kind of textbook sequence here starting first, what we see above here, what we see here with IgMs, and then later on the onset of IgGs. Basically, what we see is that the IgGs are coming right after the IgMs, and sometimes even after, sometimes already before the IgM's onset. So this is basically not really a good marker for an indicator of an early infection when it comes to IgM, which is normally used as such, giving textbook knowledge. And it's also important to keep in mind regarding the use case also for the test. And when we look at the different antibody levels, there's also not sufficient evidence that they correlate with the clinical severity in order to really be able to use them as a diagnostic for severity. There are certain publications showing a certain correlation, but this is clearly nothing that would allow an antibody test here a quantitative antibody test to really predict the severity of the infection that a patient is undergoing. So now let me introduce you to our thoughts how we selected a target structure for our antibody assay. The development of the anti-SARS-CoV-2 was done in a rather short time with teams really working 24/7. And we started with a large number of prototypes developed and evaluated in parallel to be faster. And then we selected obviously the best one, which laid the basis for the full development of the assay. So every antibody test needs an antigen to detect the antibodies in the samples. And as mentioned by Wim before, SARS-CoV-2 has 4 structural proteins that can in principle be used as antigens in an antibody assay. So what we did, we developed a large number of prototypes on the basis of structural proteins. And we used different configurations, including synthetic or recombinant antigens from either E. coli or eukaryotic expression systems in order to find the best candidate. As a result of the evaluation of the prototype assays, we used the nucleocapsid protein as antigen as the target for our antibody assay. The decision was made on the basis of experimental data, as well as also manufacturability. You have to keep in mind that we wanted to go from the lab scale to mass production in a very short time frame. So we needed an antigen that we could manufacture also in large amount very quickly. Looking at the performance, the nucleocapsid-based antibody assay performed better in terms of specificity. And internally, we selected specificity as the key performance driver to compare the different prototypes. So the N-based prototypes performed better than all other targets in early experiments. Another important factor was also, as mentioned, that it could be brought to large-scale production in a fast and reliable manner, because, as we all know, these assays are needed really at the spot, and we couldn't waste a lot of time for developing and for upscaling. So these aspects together led to the decision to move forward with this nucleocapsid-based antibody assay. So coming back to the main purpose that Wim also mentioned in his presentation, namely for epidemiological screening, with the aim to correctly identify all patients that have been exposed to SARS-CoV-2 to better understand the prevalence, the epidemiology, and also the dynamics of the virus, and of course, as a consequence, COVID-19. For this purpose, we defined that a high positive predictive value would be absolutely essential. On a side note, given that the levels and the chronological order of IgM and IgG antibodies appearance are variable, we thought that targeting both antibodies simultaneously following a total antibody approach, so it's not a decisive anti-IgM or IgG, it's really a total antibody approach, that may be beneficial for increasing also the sensitivity of a test. Although I have to mention this, it was not a primary focus. When we developed this test, the primary focus was clearly the specificity. Now let's have a look at the potential targets for the assay. I already mentioned the IgM, the IgG dilemma. So looking at the main antibody types, when we see the early infection, then we have antibodies that do not really effectively recognize the virus. The immune system still is in a kind of learning phase. And in this early reaction, we see IgMs, we see early and immature IgAs, but also immature IgGs. Then the immune system gets to know the virus or the pathogen in general a bit better, and mature antibodies arise that really effectively can recognize the virus and also fight against the virus. And these are the late and the mature antibodies. And one subset of the mature antibodies are really the neutralizing antibodies that directly bind to the virus and effectively can neutralize the virus. And I'll come to that in a bit. And based on these thoughts here that we had, we came to that assay design that we found extremely important to come really with higher specificity, also to make sure to detect the mature antibodies, of course, including then the neutralizing antibodies. It's very important for me here to say that we cannot really detect the neutralizing antibodies, but we see correlates between the signal that we see and the presence of neutralizing antibody. So this is how the assay format looks like. We have decided for this double antigen sandwich assay, and the test principle is the high affinity binding of the antigens to each arm of the antibody, which is put here in green on the left. For the assay reaction, the antigen is either labeled with ruthenium or biotin, and the antibody must bind strongly to a ruthenium and to a biotin labeled antigen at the same time. So we need to really have that the reaction works both labeled antigens bound to the antibody. And for a signal in the IgG assay, the binding of the antibody to an antigen can also be weaker, what we see here on the right side, for generating a signal. And therefore, there's a higher risk also that immature antibodies bind and give a signal that are not so specific. So this is basically the thoughts, or were our thoughts behind how we set up the format of the assay in order to really achieve a very high specificity. So why is it so important to be very specific and detect predominantly the mature antibodies? I already gave you a bit of a preview, stating that the mature antibodies also include the neutralizing antibodies. And what we would like to achieve with our assay is to detect also antibodies that potentially confer protection. Wim already alluded to that. We don't have really a final proof that if there is an antibody response, that the patient is also protected, but it's an assumption at this point. And I'm saying this again, we don't need to have final evidence, but there is a lot of work currently being done in that respect, looking at immunity, especially when also considering the vaccine and vaccine production and study efforts. So coming back to the neutralizing antibodies, they can effectively deactivate the virus, as already mentioned, and stop its replication, primarily by blocking access to viral receptors that are used by the virus to enter the host cells. Neutralizing antibodies that are a subset of the mature antibodies, and the studies have shown that the presence of anti-N and anti-S antibodies really correlate with neutralization, which could, once more, evidence is available, point towards putative clinical immunity. This is important to note that we are referring here to a correlation of the positive antibody result with the presence of neutralizing antibody, as earlier mentioned, but clearly not to a direct detection of the neutralizing antibodies by our assay. This is not possible given the technology that we're using here. Another mechanism to be complete is to stop or hinder the viral replication via antibodies that interfere with the viral life cycle. Is that some antibodies bind and interfere with the viral life cycle via complex interactions within the already host cell. But as a bottom line, we could say that neutralizing antibodies may confer protection. And on a side note, looking at neutralizing antibodies and the correlation to an antibody test result is that these findings may also be helpful for the selection of convalescent plasma of already infected patients as a therapeutic for other severe COVID-19 cases. Now, talking a lot about the theory, but let's come to actual practice. We have analyzed together with the University of Geneva the correlation of the Alexis test result with the presence of neutralizing antibodies. And for this, we were referencing to neutralization experiments done at the Geneva University and did an agreement analysis of the presence of neutralizing antibodies with a positive Alexis test result. And we came to what I consider a very encouraging result, having a percent positive agreement of 86.7% and a percent negative agreement of 100% and an overall agreement of 87%. What I consider especially important is that we do not have any false positive results in the Alexis assay that would suggest the presence of neutralizing antibodies, and of course would then imply further consequence for the patient. And these results have been submitted for publication, and in the meantime there are also various other publications out that support these findings. Now let's come back to the use cases. What can antibody detection tell us? So serological assays that accurately assess prior infection to SARS-CoV-2 will be essential for epidemiological studies, ongoing surveillance, and vaccine studies that are becoming now really very important and are also currently on our doorsteps for characterizing the vaccinees according to the previous infection. So you want to know whether a patient is really SARS-CoV-2 naive or already has had SARS-CoV infection, and there an anti-N assay is extremely helpful. But then on the other hand, what can an antibody detection tell us? Negative results cannot exclude a SARS-CoV-2 infection, particularly among those with a recent exposure to the virus. And there have been also recent reports that antibody levels may wane very quickly and infected patients are losing their antibody positivity. In addition, even in the presence of antibodies, there is currently no conclusive evidence that people who have recovered from the COVID-19 have antibodies that really protect from a second infection. This is really something that is very close, very dear to me to tell you that we don't, once a positive test result is there is clearly no protection given, or at least we don't have any evidence for protection. We've been talking about specificity, now looking at the sensitivity, although I have to say we were not focusing in the development on sensitivity, but clearly on specificity. But of course, we investigated the sensitivity because there's also a need to correctly identify someone who was infected as having been infected, but also to avoid reporting someone who was infected that he has not been infected. So. And we're looking at the clinical sensitivity in patients with a proven past infection, and proven means in this case PCR positive. The sensitivity here is presented in a way that is different than other Elexis assays you may know. Here we show the sensitivity in relation to positive PCR, because simply we didn't have any information on a symptom onset, although we consider that this is something in the range between 1 and 3 days post-symptom onset. The data shown here are from patients that are confirmed to be infected with SARS-CoV-2 by PCR. And at certain time points in days after confirmed positivity by PCR, the patient samples were tested with the Elexis anti-SARS-CoV-2. And what you can see is an increasing, here we go, is an increasing agreement with PCR over time, which reflects the ongoing seroconversion and can also overlap with the affinity maturation aspect described earlier, as over time the antibodies become more mature. With the amount of high affinity mature antibodies increasing, we see a higher sensitivity with our assay, of course. From day 14 onwards, after positive PCR, we see a 99.5 sensitivity in these patients that we have tested. These are partially also sequential blood draws. And the one patient who was not reactive at day 14 became reactive at day 16. This was the one of the 185 in the lower line here. The experiments are still ongoing, so we are broadening our database, and more samples are being collected and tested, and will provide a more extensive set in the near future. So these findings that we generated in-house produced externally, and the effect of the antibody maturation and the high specificity of the test was shown in an external evaluation done by Public Health England. The Elexis test here is here in the upper left corner. Then we have the Architect test from Abbott. Then we have the Vitros test, and we have the Liaison test from Diasorin. What you can see here is nicely the continuous increase of seropositivity in the Roche test, what is also expected based on this highly specific design with the Elexis test. It reaches 100% sensitivity after 41 to 50 days in the time increment post-symptom onset. Note, this is post-symptom onset and not, as displayed earlier with our internal data, post-PCR. Whereas the sensitivity over time with Abbott and Vitros is varying, this really indicates a weaker specificity, and the sensitivity of the Liaison assay is in general not high at approximately 80% after 41 to 50 days post-symptom onset. So now let's leave the sensitivity and go back to the specificity. As I've already mentioned several times, the main focus for the development for this test was the specificity. We want to avoid reporting someone who was infected as having not been infected, but want also avoid reporting someone who was not infected as having been infected, especially when you consider back to work approaches, etc. So we did a very extensive evaluation of the assay. And here are the most recent data on the specificity, looking at diagnostic routine, blood donors, a common cold panel, and a coronavirus panel that we obtained before December 2019. These are the 4 for endemic coronaviruses that are causing common cold and that have been described way earlier and are not as dangerous as SARS-CoV-2. So when we look at diagnostic routine, we have now 6,305 samples tested. We got 12 reactive out of that. From the blood donors, it's a bit over 4,000. We got 9 false positives. The common cold panel gave 0 false positives. In the coronavirus panel, this was a panel consisting out of the 4 endemic corona or patients that had been infected with either one of the 4 endemic coronaviruses. That also didn't show any cross-reactivity. And having discussed also with experts in the field, this was their major concern, that our test may cross-react, or any serological test may cross-react with patients who have been infected with the common coronaviruses. And then we did some on really looking at potentially cross-reactive samples that are including viruses like EBV, like CMV. And the assumption is that these viruses may also trigger unspecific immune reactions that may then interfere with the assay, and especially, again, the specificity. And we also selected this common cold panel here at random, because we assume that also here there is a lot of patients that have respiratory diseases that may caused by viruses that may also cross-react, including also the other coronaviruses. So in the end, this was the result here. We, as already shown, the common cold panel, we had no reactives with the coronavirus. We also didn't have any reactives. And then we had other cross-reacting samples, like as mentioned, acute CMV, acute EBV, and very many other pathogens that we had been looking at. And we saw 4 false positives that in the end gave, in this very challenging panel, specificity of 99.47%. And finally, and very importantly, avoiding cross-reactivity to the 4 other endemic human coronaviruses that cause common colds is crucial, as mentioned. And to our knowledge at this point, we are the only manufacturer which tested a relatively comprehensive set of samples representing all 4 potential cross-reacting coronaviruses. Some competitors do not have any data at all, so there is no data on potential interference, and other manufacturers tested only a handful of samples and not all the viruses. Elexis is therefore well ahead of the competition, and we can confidently say that there is no cross-reactivities with antibodies against the common cold coronaviruses. We also saw these results confirmed in independent publications where Elexis was compared with evident diazorin. While all 3 assays presented with a high specificity, when you look at that, at low prevalence, the minor difference in specificity resulted in profound discrepancy of the positive predictive value, what we see here on the right. When we look at the PPV at 1%, the PPVs of Roche and diazorin, so the liaison and the Elexis, there is not even the overlap of the 95% confidence interval. And the architect is at 50%, and this is basically like really flipping a coin. Having touched this already now, I would like to go really into an example showing what prevalence really can cause in terms of the positive predictive value. Due to the prevalence, a positive test result in New York with a high prevalence may be very different from a positive test result in Santa Clara in California with a lower prevalence. The same can be extrapolated to distinct populations with different exposure, for example, an infectious disease healthcare worker who is exposed to a blood donor who respects confinement measures and had only little exposure to the virus. The performance parameter we would look at in this case is the positive predictive value, which takes into account not only the specificity of a test, but also the local prevalence of the disease. When we look now at these concrete examples, New York City has a reported prevalence for SARS-CoV-2 of 15%. Imagine we use an immunoassay, which we are calling, let's say, an average competitor immunoassay that has a specificity of 98.8%. You remember the Lexis SARS-CoV-2 has a specificity of 99.8%. That's one percent point difference. And this means we have just 1% lower specificity than our assay. This needs to be really clear here. And if we were to test randomly 100,000 people in New York with a prevalence of 15%, it means 15,000 of those people will be infected and should give a true positive result. If we look at 85,000, which are not infected, we would also get 1,011 false positive results out of that because of the specificity. This results in a positive predictive value of 94%. And the PPV describes the probability which a positive antibody result is a truly positive result. If you do the same with an Lexis test and the specificity of 99.8, then we would see 161 false positives in the same population, resulting in a PPV of 99%. So there's a difference, but it's not so huge as both assays have a PPV that is still above 90%, and that is acceptable. But now what happens when we look at a population with a much lower prevalence? I was pulling up the city of Santa Clara in California, which has been reported to have a prevalence of roughly 1.5%, which means 10-fold lower than New York City, using the same two assays. Again, if we randomly test 100,000 people, the absolute number of false positives does not change that much, resulting in 1,172. But now the problem is that because there is low prevalence, only 1,500 are true positives, and in combination, this results in a really low PPV of 56%. This means with the assay, in this setting, you almost have a 50-50 chance of having a true or false positive result. Again, this is something like flipping a coin. When we go now back to the Lexis assay with a 1% higher specificity, due to a low number of false positives in relation to the low number of true positives, this will result in a PPV of 89%. This is almost 90, and again, as said previously, this is acceptable, and this is a number you really can work with. And here you can see, based on this example, that even a small difference in specificity can result in a huge difference in PPV, especially in the low prevalence settings. So let's come to the summary. We can call our assay fit for purpose, given the data that I just introduced to you. But then again, what is the purpose at this point? We explained the use cases for antibody assays against SARS-CoV-2. So the primary unmet medical needs identified are the need for an assay which assesses past exposure to SARS-CoV-2 for these principal use cases of seroprevalence screening. That helps also guiding confinement measures, etc. And also for, and again, I have to emphasize this, the potential return to work program, or just telling somebody who is, whether he's protected or immune. This is something that still needs to be investigated and determined, but this would be, of course, also a potential use case once this information is available. And the Lexis anti-SARS-CoV-2 was designed in a way to detect preferentially high affinity mature antibodies and has less risk of detecting weakly cross-reacting antibodies. It was of highest importance when developing this assay, and this puts us in contrast to the indirect IgG assays, as I showed on the mechanism slide, which have a higher risk of detecting immature IgGs and low affinity antibodies that could have been generated against one or the other endemic common cold coronaviruses. And with that, I would jump to the last slide, leaving you with some questions. And not just you, these questions are also something that we ask ourselves. Do antibodies confer reliable immunity? How long do antibodies confer immunity? Another important question, how long is a patient infective? And from when on is a patient protected? And with that, with these questions that are still need to be investigated, or they are being investigated but need to be clarified, I would thank you for your attention and give back to Anafi.
Thank you very much, both, for the wonderful and very informative presentations. So we quickly jump into questions, keep them coming into the chat box. And the first one comes from Michael, and he says there are study findings that are pointing to brain damage among recovered patients. Any comment on these findings?
In respect to detecting asymptomatic patients, for example, this would be something that we are currently also considering as a potential use case, because we see now more and more, and this was also what Wim mentioned earlier, we know the virus now for a bit more than half a year. And this virus is a multifaceted pathogen that we slowly start to understand. And given the brain damage, this is something where we don't have really a clear answer why it happens now. There is also discussion currently why or how the virus can travel to the brain. But given the also potential application of the assay, this is something, since there are a lot of asymptomatic patients also that don't really realize that they were infected. This would be an application in case some odd things happen, like, for example, the brain damage, or what we also learned now from the US, largely the onset of Kawasaki disease that may also be caused by the virus. This is something where we also could post-hoc diagnose the patients, and also then, once we know more about the pathologies, really target the management of the patients accordingly, knowing that this may have been caused by the virus.
Thank you. All right. There are questions, I think, almost similar, requesting for data. Do you have performance data with regards to duration of infection? I think you touched that a bit. And then also on prevalence of mutations.
For the last question, I have to say no. We don't have. We didn't look at any mutations now.
Mm-hmm.
And for the first question, actually, we didn't have so much data until now. I mean, there are a lot of publications now coming out looking at antibody persistence, for example, in patients that already have been exposed for a longer time with a virus. And there is pretty much unison findings that the antibody levels are waning. For some, they are waning more drastically. For some, they are kind of keeping a plateau, but slowly are going down.
I see. All right.
Thanks, Matthias. I want to quickly jump in for the question about prevalence of mutations. It's very little known about the prevalence of mutation, but I think it's important to also recognize that we have also very little information about prevalence of infection overall. So the non-mutated, right? At this point in time, we have so little data about seroprevalence in populations in Africa, in Europe, etc., that I would say that is a data set that is needed earlier and more urgently than looking at mutations which may be rare or non-existent. Understanding seroprevalence in African countries or in populations in general is a very important next step also to potentially inform future lockdown measures, etc. And there we have super little evidence as well.
Right.
Thank you very much. I think moving on, there's this one on, have you developed the antibody neutralization assays as part of your R&D to confirm that indeed those antibodies inhibit the virus?
No, we had external partners here. These were largely university partners that already have established that, and we thought that that speed would be really of utmost importance here in the characterization of our assay, so that we didn't spend any time on developing our in-house assays, but reached out to our university partners that we already have to help us here, and we had collaborations with some major university centers.
Thank you. Any updates on regulatory approvals or intended applications?
So I can take that question. Yeah, so we have, for this assay, we have CE-IVD and FDA approval for emergency use authorization. And the WHO pre-qualification is not really applicable for emergency use within WHO supported countries. However, there is the WHO process of evaluating and approving emergency use, and that is ongoing and submitted to WHO.
Thank you very much. An extra question, were there any further investigations conducted on the false positive blood donors or those presenting for routine testing?
Normally, we do investigate every false positive that we get notified and just see in how far our test may have a flaw there. But this is something that is not in the extraordinary. This is something that we normally do.
Okay, all right.
I have also seen one question about asymptomatic patients that came in through the chat.
Yeah.
So I think the question, or you want to read it?
Can the expertise be used, and this was from Tufarisa Musa. He says, can the eclysis be used in asymptomatic subjects?
Yes, of course it can. What we saw with asymptomatic patients, that there may be, of course it's not a rule, but we see a large proportion of patients presenting with rather low antibody levels. But of course it can be used in asymptomatic patients, and it's also positive then.
Okay.
It will be also a major tool for seroprevalence surveys, and it's currently being used also in studies where patients are being, or where subjects are being picked randomly from the population. And in order to come up with really what Wim already had mentioned, with an estimate on the seroprevalence in general, and not just seroprevalence that is based on symptomatic cases.
All right. As we quickly round up, can we take this last question from Neka, who says, how was the result for the positive blood donor, 9 positives interpreted?
Actually, we interpreted it as a false positive that went down into our specificity calculation. But we didn't have any further explanation on that. We just sometimes have to take the results as they are. And we also have to always consider where the cut, because we have a cutoff, and when it's slightly above the cutoff, and some of them were slightly above the cutoff, then this is just owed to the setting in the terms of specificity and sensitivity. And this is something that we cannot, well, this is what comes with a test. And I think with a 99.8% specificity, this is something that happens. But nevertheless, whenever there are any positive, false positive tests coming in, then we clearly will also investigate those.
All right. No, thank you very much, both. Just quickly, in a few seconds, just a few closing remarks, 15 seconds, 20 seconds. Wim?
Okay, from us. Yeah, so closing remarks, 15 seconds. First of all, thank you very much. And second, please hang in there. I think it's a difficult time. I can see that we need to identify and understand much better what's going on with regards to COVID-19, with regards to prevalence within the community. And if you have any questions, please reach out to your local Roche representatives. We're here to help and support you where possible.
Thank you. Matthias?
Yeah, I basically can add to Wim. First of all, thanks for your attention. And yeah, I think we're on a journey that is not as nice. But looking at what we can do right now with the assays that have been coming, that have been are still in planning, I think this is something where we could contribute at least to help better manage this terrible pandemic and hopefully get rid of this virus soon.
All right. Thank you both. And for me, what's left is for me to thank everyone who joined today. I think the only announcement that we have is I think we will continue to have these sessions. I think we're going to have the next one next Wednesday, and we will be looking at a country experience, possibly from the epicenter of the COVID-19 pandemic right now. So watch out for the next advert for the session. We shall be sending it soon. We are having a bit of a challenge with our connectivity at the headquarters. So we have delayed a bit in sharing with you the slides, as well as the codes for you to be able to go to the Academy and download the certificates for the current sessions. But those that are there, you can continue and moving forward. But we are hoping that that is rectified soon after the unrest is already sorted out. With that, I would like to say goodbye and thank you for joining as usual.
Thank you very much.
Thank you.